Welcome to Biology Made Easy. In today's lesson, we are discussing the results of independent assortment. Now, the result of independent assortment can be calculated using this formula and then this formula. This formula, 2n, where n is the haploid number of chromosomes, is used to calculate the types of different gametes formed in meiosis due to independent assortment. So to calculate the different types of gametes that are formed, this is the formula that has been proposed to be used. And then this formula is used to calculate the number of possible chromosome combinations in a single gamete, also due to independent assortment. Now, in this lesson, we want to prove that the formula 2n works only when the body cell has heterozygous allele pairs. That is, if this is a body cell, this is a heterozygote, it has gene big A and small a, and this homologous chromosome also have gene big D and small d. So this is heterozygote allele pairs. So this formula works with the state of body cell. And then this formula also works if crossing over does not occur or crossing over is not taken into account. For instance, if we have this body cell with this chromosome having gene big A at one locus and gene big B at another locus, and its homologue has gene small a and small b, if crossing over does not take place between these genes, then this formula works. But if crossing over takes place between any of these alleles, this formula does not work. Then also, if the cell has homozygous alleles, like here, you have big A, in this homologous chromosome, you have the same gene big A and big A. Then this homologous chromosome, you have the same big D. This is a body cell in a homozygous allele pairs. This formula will not be able to work telling us the different types of gametes. And this is what we want to prove in this lesson. So we begin looking at some basics. What is independent assortment? Independent assortment means one of each homologous chromosome go into a gamete irrespective of which one of the other homologous chromosome goes into the same gamete. It occurs at meiosis, specifically at metaphase 1, ending at anaphase 1. What I have said simply means that independent assortment means this is a homologous chromosome, this is a body cell, this is a homologous chromosome. What is a homologous chromosome? A pair of chromosomes carrying similar genes. One of the chromosomes is from mother, maternal. The other chromosome is paternal. This is a homologous chromosome. This is also another homologous chromosome. And independent assortment is telling us that in the formation of gametes from this body cell, this chromosome of this homologous pair goes into a gamete irrespective of which chromosome is going from the other homologous pair. So this chromosome can go into a gamete with this chromosome, or this chromosome can go into a gamete with that chromosome, and this chromosome can go into a gamete with this chromosome, and this chromosome can go into a gamete with this chromosome, or this chromosome can go into a gamete with that chromosome. This is what the law of independent assortment is telling us. That the homologous chromosomes sort out independently of other homologous chromosomes. Now, this is a body cell with how many chromosomes? 1, 2, 3, 4. So it has 4 chromosomes, but 2 homologous chromosomes. The 4 chromosomes, they are paired up paternal, maternal, paternal as homologous chromosomes. So 2 homologous chromosomes. Now the chromosomes in this cell, the diploid number, that is all the chromosomes, this 2n is 4. But only half the number go into a gamete. And the number that goes into the gamete is haploid number, that is n. Now, the formula 2n. So the n is the haploid number of chromosomes. That is the formula to calculate the different types of gametes that result from independent assortment. And what I found is that when the body cell has heterozygous allele pairs, then this formula works to tell us the number of different gametes that could be formed. So this one, individual body cell, has this heterozygous pairs. It has gene big A, small a. This is a homologous pair. It has another homologous pair, big D, small d. This is a heterozygous allele pair individual. Now, you know, before meiosis takes place, as the cell prepares to divide, the DNA replicates, and so this chromosome becomes 2 sister chromatids. This one, two sister chromatids. So this one is what is going to go through meiosis and produce gametes. The process that goes on here, we will discuss in detail in our next lesson. But we can work out the gametes that can come out of this individual just by using this. We know that this individual will separate this homologous chromosome. Big A can go with big D to form the gametes. Big A can also go with small d to form a gamete. Small a can go with big D into a gamete. Small a can go with small d into a gamete. So here we have 4 different types of gametes. And these 4 different types of gametes, using this formula, we would see that it agrees. When these are heterozygous allele pairs in the body cell, we can have 4 different types of gametes. So if we use this formula to do this, you know the 2, the n, you know you have 4 diploid number of 4, 1, 2, 3, 4, haploid number of 2, so n is 2. So you have 2 times 2, and you have 4 different types of gametes. It works instead of crisscrossing. But when you have a homozygous state, a homozygous body cell, this formula will not work, because in a homozygous body cell, the type of gametes that can be formed, you know, this chromosome can go into a gamete with this chromosome. This chromosome can go into a gamete with this chromosome. This chromosome can go into a gamete with this chromosome. This chromosome can go into a gamete with this chromosome. So all the different types of gametes that could be formed from this homozygous alleles is one type of gamete. So if we are using the formula 2n, and of course this organism, the diploid number is 4 because it has 4 chromosomes, 1, 2, 3, 4. The haploid number is 2, right? Half the number of chromosomes is 2. So for this individual, we can have 2, which is 4. So it's supposed to produce 4 different types of gametes. But you see that in actual fact, it's only one type of gamete that can be produced in the homozygous state. So this formula works only when one is considering individuals with heterozygous pairs of alleles. So if we have an individual that has heterozygous pairs of alleles, and then you also have homozygous chromosomes, to work out the different type of gametes, the n, consider the number of heterozygous pairs. So in this case, we have 1, 2, 3 pairs. So the n should take the heterozygous pairs which is 3. So the number of different types of gametes that could be formed, it will be rather 2 raised to the power 3 when n is 3, and so that will be 8. 2 times 2 times 2. And so with this, we can have 8 different types of gametes, the homozygous alleles. Now we want to discuss the formula 2n, with n the haploid number, that makes us work out the number of different types of gametes that are formed due to independent assortment, meiosis. Now we are discussing where there's either no crossing over or crossing over. So we have a body cell like this. We have gene A, gene B on the same chromosome, and its homologue has small a, small b. We have the other homologous chromosome. This chromosome has big D with big E, and its other chromosome has small d, small e. So this is a situation where a number of genes are coming in. You see that it's still heterozygous pairs. And this, when no crossing over has taken place, the number of different types of gametes that will be formed will agree with this formula. So you know these chromosomes, and we have sister chromatids, sister chromatids, sister chromatids, sister chromatids. But because there's no crossing over, we can easily cross the type of gametes that could be formed just using this. In our next lesson, we'll do the detail where these chromosomes, they align at the equator at metaphase 1. So the type of gametes that could be formed with this, we can have this chromosome going into the same gamete with this chromosome. So we can have A, B here going with D, E. This is one gamete. We can also have the same AB going with this chromosome. We can have this chromosome AB going with this. And then we can have this chromosome, this one, going with this as one gamete. And we can also have this chromosome going with this chromosome as one gamete. So when there are a number of heterozygous allele pairs, plenty even on the same chromosomes, and there's no crossing over among the non-sister chromatids, no crossing over, then the formula 2n works. This, you have 4 different types of gametes formed. You have these 4 chromosomes with a number of alleles or genes on them, 4 chromosomes, diploid number of 4, haploid number of 2. Despite the fact that there are so many genes or alleles, once there's no crossing over and the alleles are heterozygous allele pairs, we'll have 4 different types of gametes agreeing with this formula. Crossing over can occur between non-sister chromatids of homologous chromosomes. So we can have crossing over between here, and then we'll have the situation where we have this chromatid and we have this, instead of this becoming big B, it becomes small b here, you have crossing over, and this one too becomes here, instead of small b here, you have big B here. And so here, there's crossing over between non-sister chromatids of homologous chromosomes. There can also be crossing over here between non-sister chromatids of this homologous chromosome. And when this happens, we have a situation like this, where you have this chromatid is like this, but this chromatid, you have big D here, small e here, where the crossing over took place. Here you have small d here and big E here, where the crossing over took place. So when you have crossing over taking place, the formula to work out the number of different gametes, this equation does not hold. And that's what we want to show here. You know, we'll look at the detail of what is happening at metaphase in our next lesson, but the long and short of it is that at anaphase 2, this centromere will break, and so we'll have these chromatids that will go into various poles. We can easily do this by just looking at this. This is a homologous chromosome, this one, this one, this one. Now, this chromosome can enter a gamete with this chromosome, this AB. This AB can go into one gamete with this DE. This same AB can go into one gamete with this one. This AB can go into one gamete with this one. This AB can go into one gamete with this. This same AB can go into one gamete with this one. We can pick this AB, which is going into one gamete with this one, and we can do that for all of them. It's easy if we just draw a Punnett square and fit the chromosomes in and get the gametes. So we draw a Punnett square like this. These are chromosomes. Chromosomes. So we have this chromosome AB here, this chromosome AB here, this other chromosome here, this chromosome here, this chromosome here. Well, so this chromosome can go into the same gamete with this chromosome. So we have these chromosomes on this line. We have this chromosome. So this chromosome, this one, can go into the same gamete with this, and this is what is here. So we have A, B, D, E here. This same chromosome can go into the same gamete with this one. And we can fill it up, and we see that the different number of gametes that we will get here is 16 different types of gametes. We'll see we have 16 different types of gametes, and it doesn't go with this equation because these are two homologous chromosomes. We have 4 chromosomes, 2 homologous chromosomes, diploid number of 4, haploid number of 2. So, in actual fact, we could have the haploid number of 2 and this 4 different types of gametes. But this will give us 16 different types of gametes. The same body cell with the same genes in heterozygous allele pairs, no crossing over. The formula 2n works. We have 4 different types of gametes. 2 diploid number of 4, haploid number of 2, and 2 raised to the power 2 is 4. But when there is crossing over between some of the genes, crossing over between non-sister chromatid of homologous pairs, we end up with 16 different types of gametes. That doesn't agree, sort out with this equation. We want to look at this formula, 2n all raised to the power 2, that determines the number of possible chromosome combinations in a single gamete cell. Now, n is a haploid number, so if we have this body cell, the haploid number is 2. Now, this equation tells us the number of possible chromosome combinations in a single gamete cell. You know we have gametes. This will give us these gametes AD, A small d, small a, big D. These are the gametes that we can have in this cell. And this is telling us that the number of possible chromosome combinations in a single gamete, you have one cell going through meiosis, we will have 4. We'll have a tetrad, 4 cells formed out of that one cell. So if we have a gamete like AD, we won't have only one AD. The possibility is to have 4 types of AD. So it means that this one where we have 4 different gametes and 2n, the number of combinations that we could have is 2n here. We have 2, the haploid number is 2 times 2, so we have what? 4 raised to the power 2. So with this homologous chromosome, we have haploid number of 2, raise all this raised to the power 2. The number of possible combinations that we have here is 16. And it's 16 because this AD, we can have 4 of these ADs. Because each cell is going to divide into 4 tetrads. So there's a possibility of having 4 of these ADs. So there's a possibility of having 4 of these ADs. There's a possibility of having 4 of this type. Possibility of having 4 of this type. Possibility of having 4 of this type. If we have 4 plus 4 plus 4 plus 4, we'll have 16 different combinations of chromosome in a single cell. Possible 16 different combinations. So we could have a combination like this where it's all AD, or we could have some mixture. So that is how this equation works, and it also will work when you are looking at heterozygous pairs and the chromosomes. It will not work when you are looking at homozygous pairs or you are considering crossing over, as we have explained. This brings us to the end of our program for today. If the lessons have been useful, give us your likes and kindly subscribe. Thank you very much. Bye-bye.